Hi, this is Rachel Sklar. Thank you so much to the committee for selecting this research for an award. It's truly an honor. I'd like to start by mentioning that any sanitation system comes with its health risks, community risk and occupational risk. And though we know of these occupational risks, there have been few occupational risk assessments along the fecal sludge management process. Many risk assessments have been conducted in the context of the application of recycled sludge for agriculture, but there's fewer studies looking at risks to workers in upstream processes, looking at risks to workers who actually collect and process the waste. And that was the purpose of this research, to elucidate the risks to sanitation workers in non-sewered sanitation systems, and in particular, resource recovery systems. We wanted to develop a sampling and analysis framework to pave the way for doing this type of risk assessment in other scenarios as well. And then with this particular study, we wanted to produce some findings to assist in planning and optimizing the sanitation process and ensuring that workers are protected at every step. So the population that this study is referring to is the workers at a municipal scale waste to fuel plant that was operating in Kigali from 2015 to 2017. We took samples from 5 tasks. Those tasks were septic tank and pit latrine emptying, the truck waste release at the waste to fuel plant, mechanical dewatering, solar drying, which involved tilling the greenhouse, tilling the sludge in a greenhouse, and thermal drying in a vertical drum dryer, which served to remove final moisture from the sludge. So the sampling for this project was done in a participatory way, which means that we trained the workers at the waste to fuel plant to perform much of the sample collection, the structured observation, and some of the sample analysis that was done for this project. So we looked at 2 exposure pathways, inhalation and ingestion. Along the inhalation route, we chose to look at endotoxin as the primary hazard because airborne endotoxin has been shown to cause health effects for workers in similar types of waste management industries, sewage treatment, composting plants, garbage handling facilities. And along the ingestion pathway, we chose to look at pathogens that have been associated with diarrheal disease, specifically in Rwanda, those that were quantifiable by qPCR, and those that had established dose response curves in the literature. And through that process of selection, we landed on Cryptosporidium and adenovirus. To measure endotoxin in the breathing zone of workers, we use personal air sampling methods to pull air onto a filter and process it for endotoxin concentration. What we can see is that once waste arrives at the plant and starts undergoing that 3-stage drying process, the endotoxin exposure increases with the decrease in the moisture of the fecal sludge being handled. That makes sense. Drier, dustier waste gets into the breathing zone more readily. And we think that both pit latrine and inlet concentrations, we think that the pit latrine workers and the inlet workers were exposed to concentrations that were so high, despite the high moisture content, because those workers get pretty close to the sludge itself and are likely exposed to the aerosolized particles at high concentrations and at very close range. And if we compare the area sample to the personal sample of the pit latrine task, that sort of supports this hypothesis. As you can see, the concentrations from area samples are an order of magnitude lower than those of the personal samples. Now, our methodology was a bit more involved to measure the health risk of ingestion ingesting the pathogens via the fecal oral route. So we developed a quantitative microbial risk assessment model to estimate the ingestion by these workers. And we did direct measurements for 3 parameters in that model. One was the concentration of indicator bacteria on worker hands and gloves. The second was the ratio of pathogens and indicator organisms in bulk sludge samples from each of the tasks. And the third was the frequency of hand-to-mouth activity during a work task. So I'll tell you about how we use those various samples. So for the bulk samples of fecal sludge, we looked at the concentration of both indicators and pathogens in the same sludge sample. So this gave us a pathogen to indicator ratio. For the hand rinse samples, we measured the concentration of indicators and then used the ratio we developed from the bulk samples of that same task to estimate pathogen concentrations on the hands and gloves of the workers in that same task. And once we had the estimates for concentrations of pathogens on hands and gloves, we estimated how much of that got ingested by incorporating factors such as the observed frequency of hand-to-mouth and hand-to-face contact, literature values for the surface area of the hand that's mouth, the transfer efficiency of the pathogens from the hands and the gloves to the lips, and the viability of the pathogens on those hands and gloves, which we made the conservative assumption that it was 100% for this study. So we measured the concentration of bacteria on gloves and ungloved hands before and after each task that a worker completed. And we divided the tasks into 2 types. Collection tasks, so those that were done emptying the fecal sludge from the latrine or the septic tank, and then facility tasks. And those tasks were done at the waste-to-fuel plant, including the dewatering, the solar dehydration in the greenhouse, and the operation of the thermal dryer. So in general, we found there to be an accumulation of bacteria on both the hands and the gloves over the course of a work task. And since there were higher concentrations after the tasks when gloves were used on the hands, this could mean that the gloves may not be working as an efficient barrier. We also learned that the concentration of bacteria on gloves was high before tasks were even completed, suggesting that maybe there was poor storage of the gloves or poor sterilization of the gloves in between tasks or in between work days. In our bulk samples of fecal sludge from the different stages in the process, we found the geometric mean of the concentration of adenovirus an order of magnitude higher in the collection samples, so in the raw sludge, than in any of the sludge samples that were taken at the facility and in the final fuel samples. We didn't have many samples with detectable crypto concentrations, and those that were within the detection limit showed pretty large concentration spread. And of the 3 task types, we did find the highest percentage of samples with crypto in the fuel. So, although the process was designed to completely remove pathogens, we have several hypotheses to explain the detection of both indicators and the 2 pathogens in the final fuel product. One is that if the sludge is going through the process as these non-uniformly sized clusters of particles, some of the sludge in the center may not completely dry. Another hypothesis is that a recolonization or recontamination event can occur at some point during the process where sludge from upstream in the process may blow over to some of the other workstations or otherwise get mechanically transferred over to that to the fuel station. Once we had all of our parameters in place for each task type, we ran a 10,000 trial Monte Carlo simulation, which was used to estimate the worker's probability of gastrointestinal disease in that task. And between the two pathogens in both task groups, adenovirus posed a higher risk of disease, and getting exposed to adenovirus during the collection activities was the highest risk scenario that we looked at. Some general recommendations are to introduce more automation in the high exposure tasks such as pit emptying and greenhouse tilling. Of course, education could also go a long way in preventing behaviors like engaging in any kind of hand-to-face or hand-to-mouth activity during work tasks. And education about proper cleaning and storage of PPE. And with regards to PPE, storing it in a place where it can't be contaminated when it's not being used is super important. Can also be part of the education program, because as we saw, storing gloves in unprotected areas within the fecal sludge to fuel factory could lead to the gloves getting dirty and contaminated before the workers even use them. So putting them in some kind of isolated box or container for storage, having a separate storage room, that could all help in ensuring that they stay clean. The PPE stays clean while it's not in use. I just wanted to say that this research is dedicated to my good friend and colleague Wellers. He was my colleague in the lab, and he was really great. And he's one of many sources of inspiration that I carry with me to remind me of the importance of not just doing the work, but how we do it, and partnering with communities to give communities a voice, and engaging in participatory research, the kind of research where we're the outside participants and communities are the leaders. Thank you so much.